You know, deep inside every single living thing, there's a clock. But it doesn't tick in seconds or minutes. No, it ticks in the most fundamental rhythm there is: life and death, growth and division. This is the incredible story of the cell cycle, a tiny molecular timer that literally decides the fate of every cell in your body.
I mean, just stop and think about that for a second. You started out as one single cell, just one. And now you're this incredibly complex person made of trillions of them. How on earth does that happen? Well, the answer is in a process that is just mind-bogglingly precise.
It's all because of the cell cycle. The best way to picture it is like a perfectly choreographed dance. A cell grows, it copies every single piece of its DNA, and then it splits into two perfect, identical copies. This orderly process is the engine of all life, all growth, all healing, and it's happening inside you millions of times as we speak.
But see, that precision is absolutely everything. The whole system is built on checks and balances, on go signals and stop signals. So, what happens if one of those signals gets stuck? What if the clock breaks?
This is what happens. A cell that completely forgets how to stop. It just divides and divides and divides again, ignoring all the rules. So yeah, this isn't just a broken clock, it's more like a runaway train. And it is the absolute fundamental basis of cancer. To understand the disease, you have to understand the clock.
Okay, so to really get a handle on this clock, we need to meet the main players, the master mechanics that make this whole system tick. Let's just call them the engine and the key.
The whole thing really boils down to a crucial partnership between two types of proteins. If you can get your head around these two, you'll understand the very heart of how your cells are controlled.
Okay, so check this out. On one side, you have these things called cyclin-dependent kinases, or CDKs. Just think of them as powerful little engines. They're always hanging around in the cell, just waiting. The catch? They're totally inactive on their own. On the other side, you have the cyclins. These are the keys. Now, their levels in the cell rise and fall on a very specific schedule. When the right cyclin key is finally made, it finds a CDK engine, it plugs in, and bam, it turns it on. The engine roars to life and pushes the cell into its next phase.
So, what does this engine actually do when it's turned on? It performs this really important chemical reaction called phosphorylation. It's basically a molecular light switch. The active CDK-cyclin pair zooms around the cell, slapping these little phosphate groups onto other proteins. This flips them on or off and gives the whole cell the green light to move forward.
So you see, the whole thing is this really elegant loop. First, the engine's just sitting there waiting. Then, the specific key needed for the next step gets made. That key turns the engine on. The engine does its job, driving things forward. And then, and this is super important, the key gets destroyed. This shuts the engine off, making absolutely sure the cell can't move ahead until the next key is made for the next step. It's a one-way street, and that's by design. All right, so now that we know the players, let's actually follow them and see how this engine and key system pilots the cell through its entire journey, from just growing all the way to splitting in two. Right, so it's not just one engine and one key. That'd be too simple. The cell actually has a whole toolbox of them. There are specific keys, or cyclins, that get the cell ready to copy its DNA, and then other ones that actually help with the copying. Each different pair acts like a guard at a checkpoint, making sure one phase is totally finished before the next one can even think about starting.
But out of all these different checkpoints, there is one master switch, the big one, the final, irreversible command that tells the cell, okay, we're doing this. It's the point of no return.
And that master switch? It's a complex called mitosis promoting factor, or MPF for short. Its behavior is tied directly to our key. You can see it right here on this graph. As the cell gears up for division, the level of its special mitosis key, the mitotic cyclin, starts to climb and climb and climb. And as more P's pop up, more MPF engines get activated. This whole process causes MPF activity to surge to this dramatic peak, which gives the final powerful push into mitosis. And what does that peak of activity actually do? Pretty much everything. When it hits that peak, MPF basically goes on a mission, phosphorylating all the key targets to orchestrate this incredible theater of mitosis. It's what makes the chromosomes condense, what makes the nuclear envelope break down, what makes the spindle fibers form. It is the absolute final go signal from our little molecular clock. Now, you can't have a powerful engine without some good brakes, right? I mean, that would be a total disaster. Well, the cell knows this. It has built-in safety systems, the emergency brakes.
Even just starting the engine has its own safety check. See, even after the cyclin key plugs into the CDK engine, it's not instantly ready to go. Another enzyme comes along and sticks a phosphate on it that actually blocks it. You can think of it like a safety chain on a door. The engine can only truly fire once a different enzyme comes and removes that safety chain. It's this really clever double-check system to make sure the engine doesn't start up by accident.
Okay, but what if something is really wrong, like the cell's DNA gets damaged? Well, the cell needs to be able to slam on the brakes and stop everything right now. And for that, it uses a whole other set of molecules.
And that's where these guys come in, the CDK inhibitors, or CKIs. If the other thing was a safety chain, these are the actual brake pads. When the cell spots a big problem, it deploys these proteins. Their job is super simple. They just physically grab onto the active engine-key combo and completely jam it up. They are the cell's ultimate emergency stop button, halting the whole cycle so repairs can be made.
Okay, we've met the engine, the key, and the brakes. We've seen just how exquisite the control system is. So now, let's talk about what happens when it all goes horribly wrong. Because this right here is the most important takeaway of all. Every single system we've just talked about, the cyclins, the CDKs, the inhibitors, these are the very things that are broken in cancer cells. Cancer, at its very core, is a disease of broken regulation.
So how does it break? Well, it can happen a few ways. Maybe a mutation causes the CDK engine to get jammed in the on position, even with no key. Or maybe the cell starts making way too many cyclin keys, so the engine is just always on. Or, and this is a really dangerous one, mutations can damage the genes for those inhibitor proteins, which is like cutting the brake lines entirely.
No matter how it happens, the end result is always the same, a cell that is completely deaf to all stop signals. Its internal clock is just totally shattered, stuck on one command, divide. It just relentlessly copies itself over and over, creating a tumor, which is really just a physical symptom of a single cell that lost its timing.
You know, when you understand this amazing clockwork, it's not just about cancer. It reveals a fundamental truth about life itself, that so much of biology is built on timing. And it kind of makes you wonder, doesn't it? If this is the tiny, invisible clock that governs our cells, what other invisible timers are ticking away inside of us, shaping who we are?